Every living cell, including those in the heart, maintains an invisible yet measurable electrical difference across its membrane, typically ranging from -70 to -90 millivolts, with the inside negative relative to the outside. This is called the resting membrane potential, the cell's hidden battery. These differences are actively created and maintained by the sodium-potassium adenosine triphosphatase pump, which uses adenosine triphosphate to pump 3 sodium ions out of the cell and 2 potassium ions into the cell, producing a small net loss of positive charge and contributing to electrical imbalance. The primary determinant of the resting membrane potential, however, is potassium diffusion. As the concentration of potassium is higher inside the cell compared to outside, it diffuses out into the extracellular space down its concentration gradient through leak channels. This movement leaves behind negatively charged intracellular proteins and phosphates, making the inside of the cell negative. The process does not continue indefinitely. As the interior becomes more negative, an electrical gradient begins to pull positive potassium ions back into the cell, along with some sodium ions. The resting potential is reached at the point of electrochemical equilibrium, where the outward force of the concentration gradient is exactly balanced by the inward electrical pull, resulting in a resting membrane potential of about -70 millivolts. The lipid bilayer is a near-perfect insulator for charged ions, so ions can only cross the membrane through specialized protein pores called ion channels, which act as gates that open and close to regulate ion flow and control essential cellular functions such as electrical signaling. These channels respond to factors such as changes in voltage, binding of chemical messengers, or mechanical stress, allowing the movement of ions such as sodium, potassium, calcium, and chloride. This process, known as gating, enables ion channels to switch between open and closed states depending on environmental conditions, where voltage-gated channels respond to changes in membrane potential, ligand-gated channels open when specific molecules bind to them, and mechanically gated channels respond to physical forces such as pressure or stretch. Before diving into the heart's special rhythms, it is useful to study the classic action potential as seen in a neuron because the heart uses the same fundamental principles. An action potential is the fundamental electrochemical spark or impulse that allows neurons to communicate. The action potential occurs through a precise sequence of ion channel activity. In the resting state, voltage-gated sodium and potassium channels are closed, and the membrane remains at about -70 millivolts due to the sodium-potassium pump and potassium leak channels. When a stimulus brings the membrane to the threshold of about -55 millivolts, voltage-gated sodium channels open rapidly, allowing sodium ions to rush into the cell and produce the sharp rise known as depolarization. At the peak of about +30 millivolts, sodium channels become inactivated while voltage-gated potassium channels open, allowing potassium ions to flow out of the cell and causing the membrane potential to fall during repolarization. Because potassium channels close slowly, the membrane potential briefly becomes more negative than the resting level, producing hyperpolarization. Finally, potassium channels close, sodium channels reset to their closed state, and the sodium-potassium pump restores the original resting conditions. Now moving on to the heart. The cardiac action potential is the basic unit of electrical activity in the heart and is responsible for producing cardiac contractions. It is a brief change in electrical voltage across the membrane of heart cells that triggers each heartbeat. Unlike the short electrical signals seen in nerves which last only a few milliseconds, cardiac action potentials are much longer, lasting about 200 to 400 milliseconds. This ensures that cardiac cells remain in an absolute refractory period, which is a state in which they cannot be restimulated. This prevents the heart muscle from undergoing a sustained fused contraction, known as tetanus. The long duration of the action potential also ensures that each contraction is followed by a necessary period of relaxation, allowing adequate time for the heart chambers to refill with blood. Not all cardiac cells behave the same. The heart contains two fundamentally different electrical cell types, fast and slow response cells. The fast response type occurs in contractile cells of the atria, ventricles, and Purkinje fibers. It is specialized for rapid conduction, ensuring synchronized contraction of the heart chambers. The slow response type is found in pacemaker cells of the sinoatrial SA and atrioventricular AV nodes. It initiates the heartbeat and regulates its rate. With this, let us first explore the fast response cell in detail, the ventricular action potential. The ventricular myocyte action potential has the classic cardiac shape, a rapid upstroke, a brief notch, a long plateau, and repolarization. It is divided into 5 phases 0 to 4. It begins with phase 0, where a stimulus opens fast voltage-gated sodium channels, causing a rapid influx of sodium ions and a sharp depolarization of the membrane potential toward a positive value. This is followed by phase 1, a brief period of initial repolarization. During this phase, sodium channels inactivate and transient outward potassium channels open, allowing a small efflux of potassium ions. Next is phase 2, the plateau phase, where calcium enters the cell through L-type calcium channels while potassium continues to leave. The balance between these opposing currents maintains a relatively stable membrane potential, enabling sustained contraction. Phase 3 is the rapid repolarization phase, in which calcium channels close and potassium efflux increases, returning the membrane potential toward its resting negative level. Finally, phase 4 represents the resting membrane potential, where the cell remains stable due to high potassium permeability and the activity of the sodium-potassium pump until the next stimulus occurs. The sinoatrial node is the natural pacemaker of the heart, firing at a rate of 60 to 100 beats per minute. Unlike regular heart muscle, the sinoatrial node does not have a stable resting potential, which allows it to fire spontaneously. This automatic activity is driven by the rhythmic opening and closing of specific ion channels across 3 distinct phases. Phase 4 is the pacemaker potential, or spontaneous depolarization, and it is the most unique feature. The cell does not have a flat resting state but instead slowly depolarizes on its own due to the funny current, allowing a slow influx of sodium ions. As the membrane potential becomes less negative, transient calcium channels open briefly, further depolarizing the cell and bringing it closer to the threshold of -40 mV. Once the potential reaches the threshold, phase 0 begins, which is the depolarization or the upstroke. Unlike skeletal muscle or ventricular cells, the sinoatrial node relies on calcium rather than sodium for this phase. Once the threshold is reached, long-lasting calcium channels open, allowing calcium ions to enter the cell rapidly and causing the membrane potential to become positive. Phase 3 is repolarization, which resets the cell for the next cycle. During this phase, calcium channels close and voltage-gated potassium channels open, allowing potassium ions to leave the cell. This outward movement of potassium brings the membrane potential back down to its most negative level, around -60 mV, preparing the cell for the next spontaneous depolarization. There is no phase 1 or phase 2, meaning there is no plateau phase as seen in ventricular muscle, and this allows the sinoatrial node to reset quickly and fire again immediately to maintain continuous rhythmic beating of the heart. The sinoatrial node and atrioventricular node generate similar slow response action potentials, but they differ in intrinsic firing rate and conduction speed due to differences in phase 4, which represents slow diastolic depolarization as the membrane potential moves toward a threshold of about -40 mV. The sinoatrial node has a steeper slope because it contains a higher concentration of hyperpolarization-activated cyclic nucleotide channels, creating a leaky membrane that allows rapid sodium influx. This shortens the time it takes to reach the depolarization threshold, so the node fires more rapidly with an intrinsic rate of 60 to 100 beats per minute. In contrast, the atrioventricular node has a flatter slope and a lower intrinsic rate of 40 to 60 beats per minute because it has fewer of these channels, resulting in a slower inward current. Similar changes in the slope of phase 4 can be observed with modulation of the autonomic nervous system, where sympathetic beta-adrenergic stimulation steepens the slope to increase heart rate, while parasympathetic acetylcholine release flattens the slope and hyperpolarizes the membrane to produce bradycardia. Sympathomimetic drugs such as noradrenaline have a similar effect by increasing the slope, while antimuscarinic drugs such as atropine also increase heart rate by blocking parasympathetic influence. Antiarrhythmic drugs are classified using the Vaughan-Williams system, which groups them based on the ion channels or receptors they affect. Each class corresponds to a specific phase of the cardiac action potential. Class 1 drugs act during phase 0 by blocking fast sodium channels. This slows the rapid upstroke of the action potential and reduces conduction velocity. Class 4 drugs act during phase 2 by inhibiting L-type calcium channels. This decreases calcium influx and slows conduction through the atrioventricular node. Class 3 drugs act during phase 3 by blocking potassium channels. This delays repolarization and prolongs the refractory period of cardiac cells. Class 2 drugs, also known as beta blockers, act during phase 4. They reduce sympathetic stimulation, which decreases the activity of funny sodium channels and T-type calcium channels. This lowers the slope of spontaneous depolarization and slows the heart rate. The final step of how the action potential leads to contraction is cardiac excitation, contraction coupling, the process that converts an electrical signal into mechanical contraction in cardiac myocytes, mainly through calcium-induced calcium release. It begins as the action potential spreads along the sarcolemma and dives into the T-tubules, carrying the signal into the cell. This electrical change opens L-type calcium channels, allowing a small but crucial influx of calcium into the cytoplasm. That initial calcium acts as a trigger, activating ryanodine receptors on the sarcoplasmic reticulum and causing a much larger release of stored calcium. As calcium floods the cytosol, it binds to troponin C, shifting tropomyosin and allowing actin and myosin to interact, which produces contraction. As the signal fades, calcium is pumped back into the sarcoplasmic reticulum by SERCA or removed from the cell via the sodium-calcium exchanger, bringing the muscle back to relaxation. Now that we understand single cells, let's see how they cooperate to create a coordinated heartbeat, the conduction system. The conduction system is the heart's electrical highway that allows the cardiac action potential to spread from cell to cell through gap junctions and coordinate a heartbeat. It begins in the SA node of the right atrium, which has the fastest automaticity and sets the rhythm, and from there the depolarization spreads across the atria through gap junctions, causing both atria to contract as the electrical wave moves from right to left. The signal then reaches the AV node, the only electrical bridge between atria and ventricles, where conduction slows significantly due to smaller cells. Fewer gap junctions, and a calcium-dependent upstroke, creating a delay of about 100 milliseconds that allows complete atrial emptying into the ventricles before ventricular contraction. From the AV node, the impulse travels rapidly through the bundle of His, then into the left and right bundle branches that carry the signal down the interventricular septum. It then spreads through the Purkinje fibers, which conduct very quickly because of their large size, abundant gap junctions, and sodium channels, allowing near simultaneous activation of the entire ventricular endocardium. Finally, the wave spreads through the ventricular myocytes from the endocardium to the epicardium, producing a coordinated contraction that moves from apex to base to efficiently eject blood upward into the great vessels. The millions of action potentials generated in the heart combine into a signal that can be recorded on the body surface as the electrocardiogram ECG. The P wave represents atrial depolarization and corresponds to the action potential upstroke in atrial myocytes. The PR segment reflects the delay at the atrioventricular AV node, where slow calcium-based action potentials create a brief pause. This appears electrically isoelectric because the ventricles have not yet been activated. The QRS complex represents ventricular depolarization, produced by the rapid phase zero of the action potential in ventricular muscle and Purkinje fibers as the impulse spreads quickly through the ventricles. The ST segment corresponds to the plateau phase of the ventricular action potential, during which all ventricular cells are depolarized, resulting in no net voltage difference and therefore an electrically flat segment. The T wave represents ventricular repolarization as phase 3 of the action potential spreads, typically from epicardium to endocardium, in the opposite direction of depolarization. Finally, the QT interval reflects the total duration of ventricular electrical activity, from the start of depolarization to the end of repolarization, encompassing both the plateau and recovery phases of the ventricular action potential. A detailed explanation of ECG changes associated with electrophysiological abnormalities of the heart, such as AV blocks and bundle branch blocks, is beyond the scope of this discussion and would require a separate, dedicated explanation of the ECG as a whole.